of diabetes. The first way we might think about it is to look at a patient with diabetes who is exposed to a drug and then characterize them physiologically, genetically, or molecularly. And this is, classic traditional pharmacogenetics, and this will allow us to potentially look at who gets adverse drug outcomes. And if by understanding who gets adverse drug outcomes, we can better target existing therapies so we can avoid those. Or we can gain insights into drug efficacy, either because of variation in pharmacokinetics or PD or pharmacodynamics, but also because by going this route, we may uncover different etiological subtypes of diabetes. The other approach is the more traditional disease genetics approach, where we take people with type 2 diabetes and we compare them with people without, and we characterize them physiologically, genetically, or molecularly. And this gains us insight into disease etiology. And by understanding disease etiology better, we may be able to come up with new drugs or subtype individual populations, or just find that we can target drugs better. So I'm going to talk first around disease etiology before moving on to the sort of traditional pharmacogenetics. And in terms of disease etiology, there's a very good example of how disease etiology alters response to drugs, and that's with monogenic diabetes that I will briefly touch on. And then I'm going to talk around how we might phenotypically and predominantly genotypically today derive subgroups of patients with type 2 diabetes and think about how that impacts on response to drugs. Then I'm going to swing across to the pharmacogenetic angle and look at metformin response and give some insights into what we've learned around mechanism of action of metformin, look at metformin intolerance, and then look at GLP-1 receptor agonists and sulfonylurea response. So I've only had one slide on monogenic diabetes because I could give you a whole talk on monogenic diabetes, and that's not the purpose of today, but really just three little vignettes here. On the left, what we see is extreme sensitivity in terms of glucose lowering to glycoside, which is a sulfonylurea, in patients with a particular subtype of diabetes due to a mutation in the HNF1 alpha gene. So we call this HNF1 alpha MODY. These patients are exquisitely sensitive to sulfonylureas and can transition off lifelong insulin, or 20 or 30 years of insulin, onto sulfonylureas. In the middle is glucokinase MODY, and glucokinase is the glucose sensor in the pancreatic beta cell. What this slide shows is that people who were on oral hypoglycemic agents who were then found to have glucokinase and taken off, and similarly people who were on insulin and were found to have glucokinase and were taken off, you can see their HbA1c does not change. So whatever treatment they were on, including insulin, has made no impact on their HbA1c, really strongly supporting the fact that if we find someone with a glucokinase mutation, they do not need treatment, nor do they benefit from treatment. And the final story really here is to do with these rare group of patients who have neonatal diabetes due to activating mutations in one or other of the KTP subunit components, so the KR6.2 or the SUR1. And what we showed is that these patients were able to come off their insulin that they'd been on since soon after birth onto high dose sulfonylureas, and 90% of patients can come off. So really highlighting that etiology does matter, and monogenic etiology really makes a big difference, and it's really important that we look for these patients. But that's really just a proof of principle around etiology. But what do we know about type 2 diabetes? Well, when a number of years ago now, and 9 years ago, we looked at the genetic architecture of drug response, and compared it to the genetic architecture of common disease and common traits. You can see common disease, we have lots of relatively common variants, variants at 20 to 50% that were really making up most of, for example, type 2 diabetes etiology. But we had a bit of a peak at these lower frequency levels for pharmacogenetics, which was driven by pharmacokinetic variants, or the very rare variants that are associated with severe ADRs. When we come to think around type 2 diabetes drug response, it's very much more likely to follow the architecture of common disease or type 2 diabetes. So we're looking for relatively small effects and probably hundreds or thousands of small effect variants that will alter response to diabetes drugs. There is a concept now generally accepted in the diabetes community, which we call the palette model of diabetes, because, and this is really from Mark McCarthy, and the concept here is that if we imagine every individual with diabetes is a different color, and the color they are depends upon how defective some of their etiological pathways are. So most people will just have a small defect in multiple pathways, potentially hundreds or thousands, and they will be a sludgy brown color. Whereas some people will be more extreme and they will have developed diabetes here because they've got a lot of defects in this red pathway, and they will be red on the palette, and others will be blue on the palette. And potentially these people, well, I think generally we'd say it's very hard to dissect out these people in the middle. There's a whole load of people with type 2 diabetes who are genetically very difficult to tease apart because the effect sizes are so small. But there are people at the extremes who aren't monogenic, but they are more extreme within this type 2 diabetes space that we might be able to identify and treat differently. And that's the purpose for this slide. So if we think around these different colors as different pathways, so beta cell function, insulin action, and the liver and the muscle, for example, then what we want to do is try and tease out these component pathways and work out where people sit in the palette. So you can do that using their phenotypes. You could measure insulin sensitivity, insulin resistance, any other measures you could find, hip measurements, thigh measurements to look at fat distribution. And that has been shown to be very helpful. And there's a whole talk I could give around the phenotypic subtyping of diabetes. You could look at intermediate measures like metabolites and gene expression, or you could look at the genotype. And I'm just going to focus on the genotype and what we could do with that to tease out these different etiologies. So one of the latest diabetes genetics, type 2 diabetes genetics papers here from Anuba Mahajan in 2018, which was a case control of about 1 million people, found over 400 diabetes associated risk variants. Within that paper, what Anuba did was also look to ascribe function to many of these SNPs and group them together according to their pathophysiological role. So you can see that there is a group of these variants that's associated with beta cell dysfunction. In fact, there's 2 different discrete clusters of beta cell dysfunction that I'll talk about in a minute. There's groups of SNPs that are associated with impaired insulin action, groups associated with adiposity. So what you can start to see is that once you've identified all of these genetic variants, you can start to say whether somebody has a predominantly diabetes because they've got these SNPs or because they've got these SNPs, or whether or not actually they're just in the middle. So when you then look at these different genetic clusters, and this is actually a parallel study that was done by Miriam Mudler and Jose Flores that was published at a similar time, using slightly different SNPs, slightly different phenotypes, but ending up with very much the same genetic clusters. We have 2 clusters on the left that are beta cell deficiency clusters, one of which has a high proinsulin and one of which has a low proinsulin. And that's important for the next couple of slides. And then we have 3 genetic clusters that are insulin resistant clusters largely. One insulin resistant, classic insulin resistance with obesity. One, a lipodystrophic insulin resistance. So they have a low BMI, a low waist circumference, and a very low HDL, in contrast to the obesity subtype. And then a group who have abnormal lipids, particularly low triglycerides despite being insulin resistant. So 3 different insulin resistances and 2 different beta cell dysfunctions. What Adam Dawud has done with Anuba Mahajan is look at these, what we call partitioned polygenic scores. So look at all the SNPs that sum up to make each of these different genetic subtypes, and then look to see how they impact on response. And we've looked here at GLP-1 receptor agonist response, and we have access to a number of clinical trial data sets. We have the Harmony phase 3 and Harmony outcome studies here. And I won't go through the detail, but what we can see on the left is the beta cell dysfunction partitioned polygenic risk score that has the high proinsulin. So, and the higher the quartile, the higher the genetic risk, the lower the beta cell function. So the people with worse beta cell dysfunction but with high proinsulin are not responding so well to GLP-1 receptor agonists. And that's about a 0.8% HbA1c reduction versus a 1.2, 1.3% difference. So it's quite a reasonable effect difference between the lower quartile. By contrast, the other type of beta cell deficiency, so these are both genetic beta cell deficiencies, but in this other type, this is the type that has the low proinsulin. We can see that the group who've got the worst beta cell function due to low proinsulin actually respond better. So we have this contrast in the character of the beta cell dysfunction that is impacting on how our patients respond to GLP-1 receptor agonists. And you can think of the high proinsulin group as those who've got a stress beta cell, and that's classic beta cell dysfunction. And the group who've got a low proinsulin as being the unstressed beta cells, potentially even the sort of the lower beta cell mass groups. And we see a similar story when we compare two different types of insulin resistance. So this is the classic obesity-related insulin resistance, where if you're more obese and more genetically obese and insulin resistant, you have a worse response to GLP-1 receptor agonists. But if you're more lipodystrophic genetically, so insulin resistance with a low BMI, then you respond better to GLP-1 receptor agonists. And you could start to see how we could use the genotype here to help decide on which is likely to be the best drug. So I'm going to move away from etiology now and look at our traditional genetic determinants of response to drug treatment. And I'm going to focus initially on metformin. So we have now done GWAS on relatively large size populations, and we're about 25,000 patients who we've now undertaken a meta-GWAS on as part of the Metformin Genetics Consortium. And this is the Manhattan plot for that GWAS, and you can see there is one signal that rises up high above everything else, and this signal is at this locus that includes the GLUT2 gene, or SLC20A2. And our SNP you can see, is fairly consistent across all the populations we look at, including the non-European populations, and has a modest effect size. So you compare the people who are CC for our response SNP compared to TT, we're seeing differences of an HbA1c reduction of about 1.1% for the TT group, up to about 1.5, 1.6%. And that's starting to be clinically important. So that's about a dose of 550 milligrams of metformin, which is about a quarter of the maximum adult dose, or about half the effect we see when we start one of the newer drugs, DPP-4 inhibitors. So it's not a trivial genetic effect here. This is something that could be clinically useful. But what does this tell us about mechanism? The long and the short of this is we still don't know, but it's intriguing what it tells us. And I'll just briefly tell you a bit about this. So our SNP is an expression QTL. So it alters the expression of GLUT2 everywhere we look. So we can look in the liver, intestine, kidney, and islets, and it alters expression in all of those. And the people who respond better to metformin have lower GLUT2 expression. So logically, you'd think, well, maybe we're told that metformin works in the liver, and we know that GLUT2 is in the liver, so maybe this is all just to do with glucose efflux in and out of the liver. I just want to share with you a little bit of data that challenges the concept that metformin works in the liver, and then talk around how our GLUT2 SNP may be working. So this is data from a Danish group from a couple of years ago who undertook sort of gold standard euglycemic clamp studies in patients who were recently diagnosed with type 2 diabetes, randomized to metformin or placebo. So we can see here that this is the liver glucose production, endogenous glucose production. And this is after metformin and before metformin. And what we can see, in contrast to what we're always told about how metformin works, is that metformin is increasing endogenous glucose production, not lowering endogenous glucose production in these patients with type 2 diabetes. It also increases glucose disposal, RD here. So something is shifting glucose out of the blood, and the liver is probably compensating by producing more, but that's not what we've always been told. We're always told metformin works by lowering glucose production in the liver. And we've done a similar study, and I won't go into this in any detail again, but we use labeled meals, and we look at glucose production. And if you ignore the yellow bars, which is a treatment with pioglitazone, and look at metformin, again, we see increased glucose production compared to before they were taking metformin in this group of controls, non-diabetic individuals. So metformin is clearly increasing glucose clearance and increasing hepatic glucose production. And what we know, and actually has been known about for some time, is that metformin works to increase glucose uptake in the gut. So this is a PET CT scan of a person who's taking metformin, and you can see the colon is completely hot. So the avid uptake of FTG from systemic circulation into the gut. So suggesting that the gut is acting as a sort of a sump that sucks up glucose, which is an intriguing concept. Given the fact that we've found a GLUT2 variant alters metformin response, we then undertook some mouse studies with some colleagues in Finland. And again, I won't go through the detail for time, but we undertook fairly extensive phenotyping on these mice who were heterozygous null for for GLUT2, and this was whole body knockout. And we found no phenotype at all in terms of insulin sensitivity, glucose tolerance, until we put them into a PET CT scanner, which is why they went across to Finland. When we put them into a PET CT scanner, we showed that that metformin increases glucose uptake in the gut, as we'd expect, and that's the wild type individuals. But the group of mice who have heterozygous loss of function of GLUT2 have even greater uptake. So somehow our variant is increasing glucose uptake into the gut, and that may be why these patients are responding better to metformin. I say that's still very provisional data. We're still doing some more specific studies at the moment on this. Okay, so what about metformin still, but now we'll think around adverse drug effects and think around GI intolerance with metformin, which is actually very common. About 10, depending on how you define it, 10 to 20% of patients get intolerance with metformin, and of those 5 to 10% really cannot tolerate metformin at all. And what Tanya Duich showed is that when we look at people who carry loss of function variants, and actually 8% of us carry 2 loss of function variants in OCT1, and we look at drugs that potentially interact with transport via the OCT1 transporter, and these are common drugs like tricyclic antidepressants or proton pump inhibitors or verapamil, then we get essentially a drug-drug gene interaction. So if this is the odds of developing intolerance to metformin, these are the people who don't have they have normal functional OCT1 and they're not exposed to an interacting drug. Then relative to them, those who are exposed to an interacting drug have increased risk of intolerance at about 1.5-fold. The genetically vulnerable group are about 2-fold. And then if you have the interacting drug and you're genetically vulnerable, your risks of intolerance are 4-fold. We've looked in another gene that's involved in metformin transport, and this is a gene called PMAT. And we've looked at a SNP that reduces expression of PMAT. So this is in the colon and the ileum. So this is a clearly potentially important SNP for transport in the gut. And what we show is that people who carry this variant, this G allele per allele, have a 1.3 increased risk of intolerance. And you can combine that with the OCT1 and see that we get an even greater risk of intolerance when you combine everything together. And what our genetic studies show is that the reduced transport from the lumen into the enterocytes results in increased metformin exposure within the lumen, and that's probably responsible for the side effects that patients get. Now, why that is, we don't know. It may be that the increased metformin is altering serotonin uptake and increasing serotonin exposure in the gut. It might, of course, be altering the microbiome, and there's quite a lot of data now on metformin and the microbiome. There could be other effects. Okay, and then I will finish off just over the next few minutes, 5 minutes, just talking around these other drugs, GLP-1 receptor agonists and sulfonylureas, and highlight how we gain insight into how these drugs work using GWAS. So this was just out a few months ago in Diabetes Care, and what we've done here is the first sulfonylurea response GWAS, and we've done this across a number of populations shown here on the left, totaling about 5,500 patients. And we look at the HbA1c change from baseline to treatment within that first year. We show that using these common variants, the SNP-based heritability is about 37%, which is actually very similar to metformin, and it's about half as heritable as height. So it's a relatively heritable trait, drug response in diabetes. And we show that we actually have 2 hits in the same chromosome here that are involved in glycemic response to sulfonylureas. The first hit, we're still looking at the mechanism of, and can't give you any more answers, but it's an intergenic SNP around this gene GXYLT1, and it's actually an EQTL, a strong EQTL for this gene in the islets, where the C allele is associated with worse glycemic response and increased expression of this gene, and this gene potentially is involved in Notch signaling and impaired beta cell function mass, and that's the bit of the story we're still trying to unpick. But an easier story to explain is the second hit, which is in this transporter SLC01B1, which some of you may well have heard of before, is a very common transporter. So this is our SLC01B1 signal, shown here in our regional association plot. And you can see a number of genes in the region, including SLC01B1, SLC01B3, SLC01B7. And again, on the right, you can see the forest plot showing that we have a pretty consistent result. And what we show is that our SNP is an intronic SNP, and it's a cis EQTL to SLC01B1 in the liver. So it's not a coding SNP altering function, it's a expression SNP altering expression of this gene in the liver. What Kathy Giacomini and Sitwa Yi did was then undertake some expression studies. They expressed these channels. So the gene is SLC01B1, the transporter is OP1B1. They expressed these in oocytes and looked to see whether or not they transport sulfonylureas. And that's exactly what we show. The SLC01B1 and B3 transport glyburide here, or glipizide, and that's inhibited by transporter, and rifampicin that inhibits the transporters. And they also showed, and I won't go on to show this data, but they also showed that statins, which are a classic well-known substrate of SLC01B1, statins interfere or inhibit the uptake of sulfonylureas across this transporter. And what we show in our paper is that when we then go back to our data and look at statin exposure, we do show a statin sulfonylurea genotype interaction consistent with this functional data. So what's the bottom line here? Well, we have a transporter, SLC01B1, where reduced expression reduces uptake into the hepatocytes. We know that sulfonylureas are metabolized by CYP2C9, and we've reported previously that loss of function of these CYP2C9 alleles reduces efficacy, increases efficacy of sulfonylureas. When we combine them together, we actually find that it's this transporter here that's rate limiting. And I won't go into any more detail here because of time. So hopefully, you can see, the final story is back to GLP-1s, and I'm not going to spend any more time on this because I am aware that I'm running short of time, but what we show is that there's this variant in the GLP-1 receptor that which is a known functional variant in the GLP-1 receptor, reduces glycemic efficacy to GLP-1 receptor agonists, but only by a small amount. But we show when we do a gene-based burden test across the genome, looking at rare variants, we find an excess of rare variants in this gene called arrestin beta 1 that's involved in GLP-1 receptor agonist response. And that effect size, that these are relatively rare variants in Europeans, but these variants are much more common in Latinos, so we may expect this to explain a much greater response to GLP-1 receptor agonists in Latinos. And when we combine our arrestin beta 1 variants with the GLP-1 receptor variants, you can see that we're explaining about a 0.3% difference, or about an overall third of the overall effect of albiglutide, which is what we're looking at here, a GLP-1 receptor agonist. About a third of that response can be explained by these genetic variants. So, in summary of pharmacogenetics in relation to diabetes treatment, I've shown genetic variation in pharmacokinetics matters. So we've described variants in transporters, in metabolism and transport for sulfonylureas, and I haven't talked today around metabolism and transport for rosiglitazone. We've shown that genetic variation in pharmacodynamic genes that are not etiological matter. So the GLUT2 variants for metformin are probably not etiological for diabetes, but clearly important for metformin. The GXYLT1 doesn't seem to be a diabetes risk variant, but is clearly altering sulfonylurea response. And the GLP-1 receptor agonist response is altered by variants in the GLP-1 receptor agonist and beta-arrestin 1. And that etiology does matter, and we have variants in diabetes risk variants that are associated with response to drugs. The classic would be TCF7L2, altering sulfonylurea response, and the partitioned polygenic risk scores that I've shown you, altering response to GLP-1 receptor agonists. I'm just going to move on and think about how does this inform decision making? Well, monogenics, it's a no-brainer. We need to look for it and it will change treatment. Type 2 diabetes, polygenics, we have moderate to large effect sizes that matter, but they only matter when we've got that genetics available, and we're not going to be genotyping people in order to decide on drug response in type 2 diabetes. But if we know it's available, then it can be incorporated with all of their other clinical information to help guide what is the best drug for an individual. And I shall stop there. Thank you very much.
Thank you very much. It was very interesting, and I hope in the future people who have decided to purchase these genetic tests and kits, and they will be able to really also use the data and analysis they have received, like 23andMe and all these kind of places where they do these